Welcome back to the Biology Domination Podcast. I am your host, and this is episode 27, Molecular Basis of Inheritance. This is genuinely one of the most conceptually rich chapters in the entire NEET syllabus. It is also one of the most rewarding because once you understand the logic of DNA, everything else clicks. Today, we are going deep. No shortcuts. We are going to talk about what the genetic material actually is, how DNA is structured, how it copies itself, how information flows from DNA to protein, and how cells regulate which genes are turned on. Grab a coffee. Let's go. Let us start at the beginning. Why is DNA the genetic material and not protein? For a long time, scientists thought proteins, with their 20 amino acid variety, would be the best candidates. DNA seemed too simple with just 4 bases. Griffith's experiment in 1928 was the first crack in that assumption. He showed that dead smooth bacteria could transform live rough bacteria. Something was passing genetic information, but he didn't know what. Avery, McLeod, and McCarty then systematically destroyed DNA, RNA, and proteins separately and showed that only destroying DNA abolished transformation. DNA was the transforming principle. The definitive proof came from Hershey and Chase. They radiolabeled the DNA of a bacteriophage with P32, a phosphorus radioisotope, and the protein coat with S35, a sulfur radioisotope. After infection of bacteria, they centrifuged the mixture. The P32 was found in the bacterial pellet, inside the cells. The S35 was in the supernatant, outside. Only DNA entered the bacteria and directed the production of new phages. That was the proof we needed. Watson-Crick's double helix model from 1953 is one of the great scientific achievements of the 20th century. They proposed that DNA is a right-handed double helix, where the 2 strands are anti-parallel. The measurements matter for NEET. The pitch, the length of one complete turn, is 3.4 nanometers. Each base pair contributes 0.34 nanometers. There are 10 base pairs per turn. The diameter is 2 nanometers. Now, let's talk about how this molecule copies itself. Meselson and Stahl settled the replication debate in 1958 using heavy nitrogen, N15. They cultured E. coli in a medium with heavy nitrogen until all the DNA was heavy. Then they switched to normal light nitrogen medium and let the bacteria divide. After 1 generation, all DNA was intermediate in density, 1 heavy strand, 1 light strand. This is semi-conservative replication. After 2 generations, half was intermediate and half was light, exactly what semi-conservative predicts. The enzymes work in order. Helicase unwinds, primase lays the primer, DNA Pol III synthesizes, DNA Pol I removes the primer, and ligase joins the Okazaki fragments on the lagging strand. A detail NEET loves. Only the lagging strand has Okazaki fragments. The leading strand is synthesized continuously. Transcription is the bridge between DNA and protein. The enzyme RNA polymerase reads the template strand of DNA, which runs 3' to 5' and produces mRNA in the 5' to 3' direction. In prokaryotes, one RNA polymerase does everything. In eukaryotes, there are 3 types. RNA polymerase I makes ribosomal RNA, polymerase II makes messenger RNA, and polymerase III makes transfer RNA and 5S RNA. In eukaryotes, the primary transcript, hnRNA, must be processed before leaving the nucleus. Introns are spliced out by spliceosomes. A 7-methylguanosine cap is added to the 5' end, and a poly-A tail of roughly 200 to 300 adenine residues is added to the 3' end. These modifications protect the mRNA and help it bind to ribosomes. Translation then occurs at ribosomes. The genetic code is a triplet code. 64 codons, 61 specify amino acids, and 3 are stop codons. The code is degenerate. Multiple codons can code for the same amino acid. It is nearly universal across all life, with a few exceptions in mitochondria. AUG is the start codon and also the only codon for methionine. Prokaryote ribosomes are 70S, eukaryote ribosomes are 80S. Gene regulation is how cells control which genes are expressed at any given time. The Lac Operon, discovered by Jacob and Monod in 1961, is the textbook example in prokaryotes. It won them the Nobel Prize. An operon is a cluster of functionally related genes with a shared promoter and operator, regulated together. The Lac Operon controls lactose metabolism in E. coli. When there is no lactose in the environment, the cell does not need the enzymes to break it down. The repressor protein, produced by the regulator gene, binds to the operator and physically blocks RNA polymerase from transcribing the structural genes. Energy conserved, no enzymes wasted. When lactose is present, a small amount gets converted to allolactose. Allolactose is the inducer. It binds to the repressor, causes a conformational change, and the repressor can no longer bind the operator. RNA polymerase is now free to transcribe LAC-Z, LAC-Y, and LAC-A, producing the enzymes needed to metabolize lactose. This is called negative regulation. The system is switched off by default and must be turned on. The Lac Operon is an inducible operon. In contrast, the tryptophan operon, the RIP operon, is repressible. It is on by default and gets switched off when tryptophan accumulates. This is the logic of metabolic economy in bacteria. And that wraps up episode 27, Molecular Basis of Inheritance. We covered the proof that DNA is the genetic material, the double helix structure and its critical measurements, semi-conservative replication, transcription and its eukaryotic processing steps, the genetic code and translation, and gene regulation through the Lac Operon. This is genuinely one of the most beautiful chapters in biology, the logic of life encoded in molecules. Episode 28 on evolution drops next. Until then, keep studying smart, not just hard. Links for the PDF notes and community are in the description. See you in episode 28.